and we're switching computers, so bear with us. Does anyone have any leftover questions while we wait?
I do.
On you go.
Hey, so Christian, awesome. Just for clarification, you do see eccentricity in the last 400,000 years in your record, or you do? Edge of it, can't tell. Okay.
And how does it square with 18 then, with benthic 18 globally? It doesn't. So what's the explanation?
The 18 is the ocean.
And it's ice volume, no? Yeah. Arguing that 18 is not ice volume?
No, it's everything ice volume.
Yeah.
Global ice volume.
We'll carry this on at the break. Thanks, Sally.
Well, hi everyone. I'm Sally Lau. I'm a postdoc at James Cook University in Australia, and I'm here going to tell you how octopus DNA can provide empirical evidence of last interglacial West Antarctic Ice Sheet collapse. So DNA is a scarf that records family history, and every animal's DNA is passed on from parents to children, and the children's DNA family history is then knitted onto DNA. So as time goes on, like thousands and thousands of years later, we just keep adding on more and more information to the DNA, and it never ends. So the DNA of anything alive today contains all the information about their ancestor. And just to illustrate the efficiency of this, for $129, if I give my DNA to 23andMe, they can tell me who and where my ancestors were for thousands of years ago. And if we do this properly and compare human DNA around the world right now, we can look at how similarly and differently the DNA has been knitted across the population, across the world. And we can use computational methods to reconstruct historical migration patterns and date the timings for it. And this is how we understand human migration in the past from Africa to Americas. We can actually use the same approach as the human migration example to test for Antarctic ice sheet collapse in the past using animal genetics, because the ancestors who have experienced this period and how they survive around it will have been knitted in the genomes. So there are 3 possible configurations of West Antarctic ice sheet in the past super warm interglacial, including last interglacial. And the first scenario is that there will be no collapse and just melt a little bit. So from an animal genetics point of view, the benthic population in the Weddell, Amundsen and Ross would be isolated from each other by the physical ice sheet. And the second scenario is that there will be a partial collapse where the benthic population would be connected by this trans-West Antarctic seaways between Weddell and Amundsen Sea. And the third scenario is that there will be a complete collapse where the benthic population will be connected by seaways between Amundsen, Ross, and Weddell Sea. But we can't actually test for this question without incorporating samples from East Antarctica, because their geographical positions allow us to test out whether the potential seaway connectivity could have been facilitated by their movement around the coast far East Antarctica. But the trouble is, it's extremely difficult to get any samples from East Antarctica. And for over 20 years, biologists knew that animal genetics carry West Antarctic ice sheet collapse signature. So we didn't dream about this one day. And many data sets so far had very low resolution data, and they also didn't have samples from East Antarctica. So that rules out the top 3. So this leaves us with the octopus Paralanitiketi as the candidate to test for this question. There are some good samples from East Antarctica, but in 2012, we analyzed microsatellite data, which is low-resolution, simple DNA sequences, and we can't date any migration pattern this way. And ideally, we want to use genomics, which is looking at DNA distributed across the whole genome, which gives us high-resolution data to utilize the most up-to-date, sophisticated genomic approach. And of course, back in 2012, it wasn't remotely possible to think about sequencing genomic data from any marine invertebrate. But technologies has improved since then, and we can do genomics on marine invertebrates now. So we revisited this question in 2018. So this Antarctic octopus has a circumpolar distribution, and for over 32 years, we gathered octopus samples all around Antarctica. We analyzed its population structure, and we find that this Tiketi has 7 genetic clusters across the Southern Ocean. So on the map here, we have the graph of their population structure being chopped up and presented spatially. So each bar here is a single sample. Each color is a genetic cluster. If there's more than one color in a single sample, that means that this sample has ancestry of multiple genetic clusters. Because not all the locations have the same color. You can see red in Weddell and blue in Ross and Dealey Land, which is a good thing because it means that this species is biogeographically structured, and this animal swims too much and makes it way too much like a fish, then any historical connectivity associated with any potential West Antarctic ice sheet collapse will get eroded by the modern contemporary migration going around the coast. So now we established that the Octopus decadi is a good candidate to test for the historical West Antarctic ice sheet collapse. We can use statistical models to test for the size and the connectivity of the population, and also measure how these features change over time. This way, I can test if there was historical migration existed through the Trans-West Antarctic Seaway in the past, alongside the modern circumpolar movement around Antarctica. So here are the steps. So the model takes on genetic data of my different octopus population. It summarizes how the DNA differ in frequency between samples and populations. Then I input a population model that I want to test. So this is an example of one of the models I tested. We look at this from top to the bottom, older events at the top, and then the present day events at the bottom. Each bar here represents a population. The gray represents the ancestor of Weddell, Amundsen, Ross, and East Antarctic population. I asked the model to consider that at a certain time in the past, the ancestor can experience a size change at time 0. Then I asked the model to consider that the Weddell, Amundsen, Ross, and East Antarctic population get split into their own population from the same ancestor at time 1. Then each of the populations here are now represented by the different colors here. Between time 1 and now, the size of the population remains the same, but there will be migration going between some of the population, as indicated by the arrows here, which the directions of the arrows here actually correspond to their movement, a clockwise movement around the coast via the Antarctic circumpolar current. The actual parameters values of time 1, time 0, strength of the migration and population size actually determined by the genetic data. My role here is actually ask the model to test these parameters, and the scenario that I lay out on this graph can explain my genetic data well. And after specifying the model, the program compares the simulation to the real data. We loop this for millions of times with many different models to see which model scenario explains the genetic data better than the others. And after 4 years, I'll end up having a converged final model that explains my real genetic data, and it can tell me if the West Antarctic ice sheet collapsed in the past or not. So for this study, I tested 17 main models involving no partial and complete West Antarctic ice sheet collapse, while thinking about their population size change and connectivity with each other over time, and also accounting for their circumpolar movement around the coast. So some models are better fitted than the others. We can compare the fits altogether statistically. So this is the following plot of the KK information criterion fit of each of the models to the real genetic data. Each file in the box plot inside represents the variation in the AIC value across replicates. So this one, which represents the complete collapse, being the best fitted amongst all the models. And if we visualize this best model on the tree, the size of bars here are proportional to the size of the population. The arrows here represent migration, and the lines on the side here represent time interval. And the brackets underneath the time represent the 95% confidence interval. So we can see that historical migration associated with the Trans-West Antarctic Seaway connecting the Weddell, Amundsen, Ross Sea began 3 million years ago, which corresponds to historical West Antarctic ice sheet collapses in the mid-Paleocene. And then the signatures of seaway migration ended at 87,000 years ago, which, considering for the confidence interval, this can only be associated with Last Interglacial. And of course, there should be other seaway opening and closure between mid-Paleocene and Last Interglacial, like MIS 31, but our models here are specifically designed to test when was the last time there was migration through the Trans-West Antarctic Seaway, which the octopus genome here shows that it was during the Last Interglacial and not in earlier geological period. So since the seaway closed up since the Last Interglacial, there was migration going around the coast clockwise and anti-clockwise until the present day. So, Last Interglacial, air temperature is 0.5 to 1.5 degrees higher than pre-industrial, which means that we're likely close to the tipping point of West Antarctic ice sheet collapse. I'm not going to get into the details of the physics here because I'm a biologist, but just 2 meters in sea level rise, 10% of global population are going to be at risk for displacement, which is terrifying. And so in this interdisciplinary study, we came across a lot of challenges. I'd like to just briefly touch on that. It's really hard to get fieldwork access across the whole Antarctica. It's an international effort. So we worked around it by using museum and bycatch samples curated over many people's careers over 32 years. This is not ideal because DNA breaks down over time, and we solved this problem by using a very new and very expensive genetic technology to put the DNA back together. And then it's understanding each other. So we work around it by visiting our geology collaborators in New Zealand, and we talk through it face to face. And then is publishing the result. So it was with the editor for 7 months because no one accepted the review request. And geneticists don't want to review a geology paper, and geologists don't want to review a genetics paper. And at this point, everyone was giving up. We decided to put it out as a preprint as a last effort to drum up some interest. So which worked, but it came with its own preprint-related challenges. Right now, I'm really happy to say that the paper is under review somewhere in a high-impact factor journal. But we don't let these challenges stop us. The power is undeniable, and I hope that I have demonstrated in this talk. So at the moment, there are 8,000 living Antarctic marine vertebrate species in the Southern Ocean. They have been trapped by and they and their ancestors have been trapped by the Antarctic circumpolar current system since 10 million years ago. And they have persisted and diversified in situ in Antarctica since. So animals respond as the environment change, and the genomes right now will have recorded how their ancestors responded to these past events in the past 10 million years. And these animals, their genome just sitting at the bottom of the ocean floor, seafloor right now, waiting for us to pick them up. And there's so many questions that we can explore, including grounding line, expansion, and retreat. If you ever need new perspective on tackling any physical science questions, just collaborate with a biologist and our lab. So you didn't get any of the genetics I've just talked about. First Stock on the Moon made a cartoon about it, and feel free to check it out. Finally, I'd like to thank my collaborators and my PhD and postdoc supervisor, Jan and Erid over there, and all the different funders, especially Australian Research Council, for supporting this project. Thank you.
Fantastic. This is like a metaphor for how instant should be carrying on. Brilliant. Questions? Yeah.
Really enjoyed your talk. Thanks. I'm just wondering, is there any particular reason that you would want to use octopus over some other benthic organism? I know I'm imagining that it might be quite difficult to capture them.
Yes.
Versus something like a sponge, which they seem to be everywhere.
So octopus being the good one out here because we need to use this model, we need to understand how they reproduce, how they move, and how does their DNA mutate over time. And for some weird reason, we are all obsessed with octopus, and we've looked into this for 30 years, so we have all this information. For other Antarctic marine vertebrate species out there, we don't actually have this kind of information to help the model. But we are moving on, like we're gonna look at more other marine invertebrates in the Southern Ocean to test with this question, because there are 8,000 of them out there, so yes.
Thanks, Sally. Right, we'll move on. Thank you. Is Keir around? Our next speaker is Keir Nichols. He's going to talk or join the dots back onto onshore